We have a few questions from the audience, and some are generic questions, which I would take first, and then closely related to your work. So without much further ado, I'll go to the first question. It's a basic question on how to design a library of inhibitors or compounds other than using computational or docking studies. Because sometimes these docking studies, when you translate into in vitro, in vivo experiments, they don't give you the same results, or that correlation, in vitro in vivo correlation, is absent. In which case, what are your, let's say, contingency plans on identifying the most potent?
Yeah, so absolutely correct, because you cannot rely completely on 100% on docking results. If you have seen the first part of our work, we have taken taurine-2 as a scaffold, which is actually proven kinase inhibitor for mTOR. And that is where the scaffold plays an important role. Suppose if a molecule is already proven to be an inhibitor for some specific target, it is much easier to derive the molecules from there. So as a starting of the lab, I also wanted to take this route of doing something which is already known, but extrapolating to something else. But if a new kinase or a new protein, you want to design a molecule, the best way is to do this blind docking with several thousands of molecules, which you can actually dock and then see what are the scaffolds which could bind to this particular protein. In that case, I would say that after you do that particular screening, you have to make some of the scaffolds in the lab. So eventually it's the experimental proof, it's much more important. So we can, instead of now you have AI tools. Okay, so it is much more reliable than the docking which used to be there 10 years before. So those tools can give you precisely of the scaffold that you would like to. But eventually the experimental detail is experimental.
Okay. You were mentioning about covalent inhibitors in the initial part of your talk. Any particular reason why acrylamide warheads are the most popular?
Oh, yeah, I missed actually explaining that particular portion. So in chemistry, it's a very simple reaction. You have cysteine in your protein, either it's a kinase or any of the protein, and you have acrylamide. There is a reaction called Michael addition. So which makes this reaction, which is like a click chemistry reaction. So it's very, very easy to do. So in the lab also it's very easy to, and the chemistry that is developed around this is very smooth reaction. So when you have a cysteine, when you have an acrylamide, it very quickly clicks. Okay, so it makes a Michael addition reaction. So because of that, the covalent inhibitors have become very popular, and it is done in vivo and in vitro very easily.
The next question, it's more related to your field. The kinase inhibitors, especially the DNA damage and repair pathway kinase inhibitors you described, do they have pan-cancer activity, or they are specific to particular cancer types?
So right now, there are no FDA-approved molecules which is specifically developed for DDR kinases, that is DNA damage and repair pathway kinases. But there are several molecules which are in the last phase of clinical trials. Okay, so those molecules actually show pan-kinase inhibitor activity, and that's why they also show a little bit of toxicity towards other toxicity that happens during chemotherapy. But most of the kinase inhibitors do show pan-cancer activity. So like imatinib, it's not only for CML. Now there are several molecules which are similar to imatinib has also shown potency against other cancer types.
So the next question was very closely related to your this answer, whether there are any other DDR pathway kinase inhibitors which are under clinical trials, and how they are similar to or different from your.
Yeah, there are several others from Vertex. So Vertex group works very lot of molecules they develop. Baylor, they also develop. So Bayer company also develop a lot of molecules towards DDR pathway. I'm crossing my figure and I'm thinking that ours will be very potent and less toxic than the ones that has been developed. But it's a long way to go. They are companies, we are academicians.
Yeah. So coming to your work on the compound J54, So it's a question related to that. Which characterization technique was used for obtaining the in vivo images of xenograft model?
This
For
is a
confirmation?
tumor, this is a basic tumor xenografts where you induce prostate cancer to mice. So Balbi mice is the one that we have used. I mean, it's done totally done in Professor Benedetti lab in Louisiana. And then you dissect that and then get this model. Yeah.
Okay. Coming to the last part related to the work in glioblastoma, was this molecule tested in vivo in animal models, or is
It's
it
been
still?
tested right now. It's been tested right now in vivo model. Yeah, we have finished the in vitro studies, so we are taking this to in vivo.
Okay. There's also a question related to GBM resistance. MGMT, which is like a DNA repair enzyme that contributes mostly to temozolomide resistance in GBM. So does this approach using K1B, address that issue as well?
Yes, absolutely. So this is my student Bhanupriya's full thesis. She has just defended her thesis. So it's not published yet much, so I have not talked about it. And maybe in some other occasion, I'll be able to talk about it much detail.
So I believe these inhibitors are actually, they act as adjuvants to chemotherapy and radiotherapy. So the idea is that it will reduce the dose or the side effects, or how is it, or both?
Yeah, it's mostly some combinatorial therapy. That's what we wanted to use it, because it will reduce definitely the dosage that is given. In case of even bicalutamide work that I was showing, the dosage that is given to normal prostate cancer patients is very, very high. But when you have J54 in combination, the dose was very, very less.
You mentioned the challenges which were there in designing kinase inhibitors. Anything specific to DDR pathway kinase inhibitors, or is it like globally for all?
The major challenge with DDR pathway kinases is their structure is just not known. So other kinases, at least the structure is particularly known. And as I told you, that very limited number of kinases have been targeted now. That is because, again, the structure is not known. So EGFR is very famous, right? EGFR inhibitors have been developed for a very, very long time. BCR-ABL for other kinases. But in case of DDR pathway, the structure, since the structure is not available, maybe now cryo-EM is flourishing. So we will probably have the structure very soon for many of the kinases. Then this would probably pick up much faster rate.
So I'll come to the final question here. What are the translational potentials of your work, or are you looking towards commercializing some of your work?
Yeah, so the first beginning stage of lab work, where we have developed a lot of strategies to express these kinases in E. coli, which is not commercially available. So for everything, we have to depend on some other company to actually supply us. So we were thinking that we have just signed up for in our incubation center and research park at IIT Gandhinagar for a startup. So which will be launched soon.
Yeah. Oh, thank you, Professor Sivapriya, for patiently answering all the questions. Do you have any final remarks to the student community and working in similar areas in general, and also specifically to the women scientists, the researchers, considering the International Women's Day is very close?
Yeah, first of all, I wish all of you guys and all of the men who actually supports these wonderful women also, because I think that's very, very important. I have fantastic support from my home, starting from my home and from my students also. The key for doing any kind of a high challenge research is to have a lot of patience. You know, when you start something very challenging, you might not get the results as much as you want. That you can see from my first part of my lab was undergoing a lot of trouble, because as a new to this area of research, students might not feel that, okay, you know, this is easily done. But that could be frustrating sometime. But if you cross that, I think you will all come out with flying colors. And also very keen noticing small things. When there is some as Professor Bertocci says, that there is no experiment which is a failure. Experiment doesn't go like you want to go, right? The experiments are always experiments. It gives you something results. So you don't need positive results. What you need is only results, and that results have to be analyzed very closely. I think if you do these two things with a lot of patience and a lot of hard work, we are ready to go.
Thank you for that wonderful message. I'm sure your journey will inspire many young researchers. With that, we have come to the close of the Q&A session. Back to you, Krishna.
Thank you, Sriranjini. And thank you so much, Professor Sivapriya. That was a wonderful message. And I'm really sure that you must have inspired a lot of our audience, to take up science more seriously. Maybe, some of the master students to pursue PhD. So thank you so much. This brings us to...
Thank you. Thanks a lot, Asha. Special thanks to you, Krishna, and the Sriranjani for making this happen. Thanks a lot.
Thank you, Professor Sivapriya. With this message, we come to the end of today's session. We hope that you really enjoyed this broadcast. I personally enjoyed it a lot. And we invite you to view the edited recordings from past events in ACS Science Talks library. They are available on YouTube, and my colleague have already posted, the link in the chat box. At the end of this session, you'll receive a very brief survey. Kindly share your feedback with us so we can continue to improve and serve you better. On behalf of all of us at ACS, we thank you for joining us today. We hope to see you again soon for more ACS events. Until then, stay safe and stay healthy. Bye-bye.
Thank you, everyone.